In this lecture, we're going to be taking a look at microbial mechanisms of pathogenicity. In other words, how do these microbes manage to make you sick? So we're going to take a look at how microbes enter you, in this case as a host, and we're going to define portals of entry. That's important. ID50 and LD50 are important terms, and we'll also take a look at how microbes adhere to host cells. So two terms here that you need to know. Pathogenicity is the ability to cause disease, and virulence is how bad it is, the degree of the pathogenicity. So here are some portals of entry. Typical portals of entry would include mucous membranes, skin. You've heard recently a lot about not touching your face with your hands. These are all portals of entry. Your mouth, your nose, your eyes, these would all be considered portals of entry. Then there's also the parenteral route. That is a needle stick, for example. So that means the microbes will be deposited directly into tissues through a needle, a hypodermic needle. Most pathogens have a preferred portal of entry, and some like to be going in as a droplet infection. Some need to have direct contact, so there's different kind of pathways. More terms here. ID50. ID stands for infectious dose for 50% of a sample population. That means how many microorganisms do you need in order to produce an infection in 50% of your sample population? It's a measure for the virulence of a microbe. If the infectious dose is extremely low, that means it really doesn't take much to get a lot of people infected. The LD50 is the lethal dose for 50% of a sample population, LD for lethal dose, and that measures how deadly is this pathogen. So how much does it take for a toxin or the pathogen to kill 50% of a sample population. So here are some examples for ID50, the infectious dose that it would take to infect 50% of a sample population. So here we're looking at an example, Bacillus anthracis, the cause of anthrax. If you're using the skin as a portal of entry, it only takes about 10 to 50 endospores. That's not much at all. By inhalation, to produce an infection, 50% of a sample population, so inhalation would take 10,000 to 20,000 endospores. And via your gastrointestinal tract, by ingestion, by eating those endospores, it would take 250,000 to 1,000,000 in order to produce an infection in 50% of your sample population. Here are some more examples for ID50s given. In this case, we're taking a look at toxins and how much it would take to produce an infection in 50% of the test population. Botulinum toxin, that's the most potent neurotoxin known to mankind. It takes an amazingly low amount to get you really sick, and that will be here 0.03 nanograms per kilogram of the person. Shiga toxin, also pretty toxic, 250 nanograms per kilogram. By contrast, staphylococcal enterotoxin, it would take 1,350 nanograms per kilogram. So these are just some examples, but botulinum toxin, Botox, ultra toxic. So let's take a look at the adherence. So that means how do pathogens attach to host tissues. And so they need to find a way to attach to your cells before they can cause problems. In the process of adherence or adhesion, that's what we're going to take a look at. So basically, what kind of structure allows a pathogen to attach to your cells. So adhesins, those are the structures, or also sometimes called ligands, on the pathogen. They bind to receptors on the host cells. So these adhesins or ligands on the pathogen could be found on the glycocalyx of the pathogen, or also sometimes on the fimbriae and other surface structures of the pathogen itself. Some microbes, they form what's called biofilms. It means these are communities of bacteria, basically masses of bacteria. They share nutrients, and they form layers where then the outer layers are exposed maybe to something, but the inner layers of pathogens are shielded from any kind of problem, and so they can live there. Biofilms, a good example would be including the plaque on your teeth. Here are some examples of how bacteria might attach to your cells or surface structures on you. On panel A, here you see how this pathogen right here is using an adhesine, a ligand. So here the surface structure right there to attach itself to a receptor on the host cell membrane. On B, here we have E. coli bacteria in yellow that are attaching themselves to the human urinary bladder cells right there. And on C, you have bacteria in purple that are attaching themselves to human skin, so on the surface of your body. Some bacteria have capsules that increase the virulence of the bacteria. So a capsule is sort of this waxy glycocalyx type of material that surrounds the cell wall, and it basically is a mechanism of disguising yourself to the immune system of the host. And it impairs phagocytosis. It's very difficult for your immune system to figure out that there is something invading you. It's sort of these bacteria, some kind of stealth fighters. They're invisible to the immune system, or very hard to figure out. I have some examples here of pathogens that use capsules to increase their virulence. Would be Streptococcus pneumoniae right here, cause of pneumonia. Haemophilus influenzae also causes pneumonia, meningitis, Bacillus anthracis, the cause of anthrax, and Yersinia pestis, the cause of plague. Here are some cell wall components that increase the virulence of pathogens. Let's start with the M protein. That's a heat-resistant and acid-resistant protein that helps pathogens that have it to attach to surfaces. It mostly helps these bacteria then to resist phagocytosis, so to not be eaten by white blood cells. Good example for a pathogen that has the M protein is Streptococcus pyogenes. Neisseria gonorrhea, that pathogen uses the OPA protein to help it attach to host cells and increase its pathogenicity. And mycobacteria, such as Mycobacterium tuberculosis, they use a waxy lipid, the mycolic acid, that is part of their cell wall, to resist the digestion by phagocytic cells. Here are some enzymes that help pathogens to increase their virulence. These are exoenzymes that mostly help pathogens to invade the host. So here will be first coagulases. They coagulate fibrinogen, basically cause clotting of blood. Kinases, they can digest fibrin clots. Hyaluronidases, they digest polysaccharides that hold cells together and help then pathogens invade the host. Collagenases, they break down collagen, which is also a binding substance that holds cells together. And IgA proteases, they destroy IgA antibodies. Here you have an image of a human heart, and you can see blood flow through the coronary arteries that are sort of imaged here for, and you can see that there's a blockage right here in this coronary artery section. In this case, the patient had an infection with bacteria, with a pathogen that produced coagulase. The clotted blood then ended up blocking this coronary artery and produced a massive heart attack. The physician treating this condition recognized that it was a pathogen that caused the clotting of the blood here in this coronary artery, and gave a shot of streptokinase, which then dissolved the clot and caused the blood flow again to go through this coronary artery. In the next image here, you see an infant with a condition called necrotizing fasciitis. In this case, the pathogen Streptococcus pyogenes produces actually streptokinase in order to dissolve clots that the body uses to limit the spread of the infection. So in this case, it works the opposite way. The streptokinase is actually helping the pathogen to invade tissues faster and then producing the tissue death that you see in this case. Here is a diagram that shows you how streptokinase works. So basically, streptokinase will digest fibrin, and then the fibrin is necessary to form the clot. And so once the fibrin is digested, the clot will dissolve. And of course, your body uses a clot in this case to limit the spread of an infection, but the streptokinase helps the pathogen to invade the area. Another trick that pathogens can use is called antigenic variation. That means the pathogen alters the surface antigens that your immune system uses to make antibodies. And once the surface antigens are different, the antibodies that your body had previously made are no longer effective. Viruses are notorious for that, and that's why you need to get a flu shot every year, because the surface antigens of the flu viruses, they change all the time, and your immune system now needs to make a new antibody. Another trick that pathogens can use is the use of invasins to penetrate into the host cell cytoskeleton. Basically, invasins, they are surface proteins produced by bacteria that end up rearranging actin filaments of the cytoskeleton. They cause something called membrane ruffling, and Shigella and Listeria, for example, they can then use the actin to move from one cell to the next and invade a larger area. Here's an image that shows you what the ruffling of host cell plasma membrane might look like. So here you have this ruffling here on the surface. In this case, it's caused by Salmonella typhimurium. And here we have another evil strategy of some pathogens to use the host's nutrients. Some bacteria, they can use Siderophores to get access to the iron supplies of the host cell, from the host cells. So iron is required for most pathogenic bacteria to grow. And in order to obtain some free iron from the host cell, some pathogens, they can produce siderophores, which are proteins that bind iron more tightly than the host cells. And so basically the pathogen rips off the host cell. Here is an example of a bacterial siderophore. It's a molecule that binds iron tightly, more tightly than the host cell proteins or molecules can do this. And so therefore, the iron is attracted to the bacterial siderophore and then helps the bacteria to grow. Some pathogens can also cause direct damage to cells, and this applies mostly to intracellular parasites such as viruses and intracellular bacteria. They usually disrupt host cell function. They use up host cell nutrients. They may produce toxic waste products, and viruses, as you know, and also some intracellular protozoa and bacteria, they multiply in the host cell and then cause the host cells to rupture and die, of course. Now let's take a look at pathogens that produce toxins to enhance their virulence and cause damage to the host. Here first some definitions. A toxin is a poisonous substance produced by a microorganism. Here's some examples given what toxins can do to you. They can produce a fever, cardiovascular problems, they can cause diarrhea or shock, and that's just some examples. There are other things. Toxagenicity is the ability of a microorganism to produce a toxin. Toxemia means that the toxin is in your blood. Emia refers to blood. And intoxication means that there is a toxin present, but without microbial growth. Let's first take a look at exotoxins. Those are proteins produced and secreted by bacteria. They're typically soluble in body fluids. They destroy host cells, inhibit metabolic functions, and cause a lot of damage. Antitoxins are antibodies that you would make against a specific exotoxin. Toxoids are inactivated exotoxins that can be used in vaccines to cause your immune system to make antibodies against a particular exotoxin. A good example for that would be tetanus toxin. We're using a toxoid to produce immunity against the tetanus toxin so that you don't have to get tetanus.